right, for this video, we're going to be looking at C1.1, which is enzymes. Our theme is interaction and interdependence. And so we're going to be focusing on how enzymes interact with other molecules. And so when we're talking about an enzyme, first we need to know what an enzyme is, which are proteins that increase chemical reaction rates, but those proteins are not changed by the reaction. They can be reused many times, and they are globular proteins that have a specific 3D site for a substrate. And that 3D site is called the active site, and the molecule that's going to bind into the active site is called the substrate. Substrates must bump into the active site to catalyze the reaction. So they're going to be randomly moving around. And then if the substrate hits the right location and hits the active site such that it can actually enter into the active site, then a reaction will happen. So if there's more bonking going on, or if there's more substrate, so there's just more concentration, then more reactions are going to happen just through random motion and more likelihood of collision. The way that an enzyme works is the model called the induced fit binding model, and interactions between the substrate and the active site change the shape of the active site. So as the substrate approaches and connects with the active site is going to change its shape to accommodate that specific substrate. However, active sites are specific to certain substrates. So not any old substrate can bind into an active site and have a reaction occur. So enzyme structure, the active site is going to apply strain onto the bonds to break down molecules or mash the molecules together to join them up to their respective components. So if this is the substrate here and the purpose is to pull it apart, there's going to be strain put on the bonds, and that's going to break that substrate into products A and B. Enzymes lower the activation energy, so how much energy is needed for the reaction to occur. And this might have double lumps in the reaction graph, but the activation energy is always going to be lower in a reaction that's being catalyzed by an enzyme. Reactions can build bonds, which would be anabolism, anabolic, which would be something like DNA synthesis, or it can break bonds, which would be catabolic. The word catabolic means breaking into smaller components. And that would be something like the digestion of food, where you're taking a big molecule of protein, for example, and breaking it into its amino acids. And so enzymes can catalyze both of those reactions, anabolic reactions and catabolic reactions. If you're having a hard time remembering the difference between anabolic and catabolic, think anabolic steroids build muscles. And so anabolic reactions are going to build bonds. And as I've said previously, the active site is specific to the substrate. So there's a few different factors that potentially could affect how your enzymes are working. Most reactions are happening in the cytoplasm where molecules are dissolved in water. So if there's more substrate, you'll see this reaction here. Just because of more collisions, there's going to be more reaction rates. And so reactions are going to occur more quickly because there's more collisions. And at a certain point, you can't maximize collisions any further. And so it's going to plateau like it has here. More heat is going to mean more movement of the molecules, which is going to mean more bonking together. And so that will also increase the reaction. But at a certain point, the enzyme is going to reach an optimum temperature. And that's where maximum bonking together is happening, but the enzyme is still functioning. At certain points, an enzyme is going to denature if you increase the temperature enough, which means that the structure of the protein itself falls apart. And once that happens, then the enzyme no longer works. It's not repairable. We just need to make new enzymes. And so once the enzyme starts to denature, then that reaction rate is going to just fall off a cliff. And so that temperature can be dependent for the enzyme. And so, for example, in the human body, where the general temperature is 36 degrees Celsius, enzymes might denature at 41, 42 degrees Celsius. whereas an enzyme for a bacteria that lives inside a hydrothermal vent, it might be adapted to deal with temperatures up to 190 degrees Celsius. And so its enzymes wouldn't denature until at a very high temperature. Lastly, pH, similar to temperature, is going to have an optimal reaction space. And so, for example, in this case, the pH of enzymatic activity is reacting optimally at pH 8. And so this isn't so much about collisions. This is about how the enzyme structure is maintained. And at a pH that's lower or a pH that's significantly higher, that enzyme can become denatured, and as a byproduct of that will stop functioning. And so it will not be able to have much activity, and the reactions will not be able to take place. Again, it depends on what enzyme we're looking at here for the pH, because normally in, for example, our bloodstream, our pH is around 8, but in our stomach, the pH is 2. So enzymes in our stomach are going to be adapted to dealing with a pH of 2 and are not going to denature in a low pH, like a pH of 2. So metabolism is the chemical reactions that are occurring in the cytoplasm of our cells, and enzymes are critical to these metabolic processes. A liver cell produces thousands of enzymes because of all of the different jobs it has, and these cells can control the chemical reactions happening by controlling the presence of enzymes. So metabolism is chemical reactions that are happening in a living organism to regulate their body processes. For example, mitochondria using glucose and oxygen to make ATP. Sometimes these reactions are cyclical, but usually they're linear chains of reactions. So 2 cyclical examples would be the Calvin cycle. This is occurring in chloroplasts. And then the citric acid cycle. This is occurring in mitochondria. And so here you can see a bunch of, for example, citrate, oxalosuccinate, succinyl-CoA, succinate, fumarate, malate, oxaloacetate. So all of these reactions are catalyzed by different enzymes. And so as this cycle is going along, then these different chemicals are going to be affected by the presence or absence of enzymes. So to summarize, enzymes cause reactions that to happen at lower energies, and these reactions may occur in multi-step processes going through a transition phase. Reactions can be sped up by using warmer temperatures or greater concentration of the reactant, and too hot or too acidic or basic will denature an enzyme, preventing it from continuing to work. All right, that's it for this video. Thanks for watching.